Hey guys, I hope you're enjoying the cold slash everybody is sick day. I want to keep us on track though for a test on Tuesday, so the first thing I know that you're going to ask me is, are we still having a test on Tuesday? The answer is yes. When we last left each other on Ash Wednesday, in each of your classes we were talking about where life could have possibly come from. And the first thing we did was said that the early atmosphere was composed of some pretty, volcanic conditions. Remember, we mentioned ammonia, NH3, methane, which is CH4. We mentioned some hydrogen gas, which is highly explosive. And we said it was very much like an early volcano, so add some hydrogen sulfide. The early atmosphere also would have had nitrogen, as well as carbon dioxide and some water vapor. This is a pretty violent, difficult, unpleasant atmosphere for anything that would have, nothing could have been alive. But it's what we call a reducing atmosphere, and it means that it's highly reactive in a particular way. A reduction is a chemical reaction, so it would result in the exchange of electrons very easily, and thus a lot of chemical reactions would happen. So the question is then, if this is what early Earth was like, where did life come from? And we said that there's a few possibilities. One, no, not aliens, but yes, life could have come from outer space, someplace else. Not TARDIS-style outer space, but we're talking about small living things arriving on Earth via a meteor or an asteroid. The problem with this idea is it's not testable. We can't really simulate that in any laboratory conditions, and if you think about it, it seems unlikely that anything living would have survived, really, from any of the violent conditions that would have arisen in a meteorite crashing to Earth. The next thing we said was there could have been a creator, but again, we can't talk about that one because how do you test it? And even if we could test it, we've all agreed that there's no wisdom in trying to test the existence of a creator. The only possibility that it leaves us then is the idea of chemical evolution. Now, I want you to be careful with the word evolution because sometimes it makes us think of what we just talked about, natural selection and Charles Darwin. That's biological evolution, which means the evolution of living things. Chemical evolution would be an increasing diversity in chemicals present on Earth. That would be caused by interactions and chemical reactions, not life. It wouldn't be something that was selected for or against. There would be no advantageous adaptations. So don't use those words that we associate with biological evolution for chemical evolution. This is simply talking about how chemicals on Earth could have reacted with each other in that icky, reducing environment of the atmosphere that we talked about. The currently accepted biological hypothesis for how that could have happened is what we call the heterotroph hypothesis, and it was developed by two scientists, Oparin and Haldane. Haldane is the one who is famous for coining that primitive soup, or that tarry ooze that we talked about, sometimes also the primordial soup model. That's where that name came from. And that describes this exact hypothesis. What Oparin and Haldane outlined is the idea that there had to be 3 steps in order for life to come about through chemical evolution. The first step would be inorganic molecules that existed in abundance. We already said there were lots of them there: ammonia, methane, hydrogen, carbon dioxide. Those inorganic molecules would have had to form monomers of our organic molecules. Those monomers are the ones you're already familiar with: monosaccharides, nucleotides, amino acids. So step 1, inorganic things had to find some way, an inorganic, non-life process to make our monomers. The second step, those monomers would have had to in some way form polymers. So we would have had to have gone from our monosaccharides to polysaccharides, from our nucleotides to our nucleic acids, from amino acids to protein. And that has to, again, normally that occurs as we know it now, as a process that living things do. But Oparin and Haldane are saying there had to be a non-living method of doing this. The third thing that they said then is polymers must be self-replicating. Self-replication, the ability to reproduce, is one of the most important characteristics of life. We don't consider things alive unless they're able to self-replicate or reproduce. This hypothesis uses that proposed reducing atmosphere we talked about, that really nasty, awful, hostile one of early Earth, to suggest that the first life on Earth was heterotrophic. So, in other words, we remember from ecology that a heterotroph is other feeding. So they're saying the very first living things fed on other biomolecules. So it fed on anything that was left over from step 2 in order to support life. For the heterotroph hypothesis, the Oparin Haldane hypothesis, to be worth discussing, we have to be able to test it. We have to be able to support assumptions 1, 2, and 3 of this hypothesis. And since probably about the 1950s, scientists have been working on doing exactly that. The first support for the heterotroph hypothesis, or Oparin Haldane hypothesis, or primitive soup model, whatever you want to call it, the inorganic to organic, the first experiment that was done by two scientists, Stanley Miller and Harold Urey. And it's a pretty famous experiment. This experiment, they simulated the conditions on early Earth in order to try and examine it for organic compounds, to see if there's any way that they could take that reducing atmosphere and they could make an organic compound. Now, notice there, I should have articulated more clearly. They could make an organic compound. So they went from inorganic to organic. And the way they did that was making this really fancy piece of glassware that you see here on the left. This piece of glassware was intended to create early Earth. So you'll notice up here in our atmospheric chamber, you can't really see it very well because I did it in white, that I circled it. They attempted to create that atmosphere of methane and ammonia and hydrogen. And then they applied electrodes to it. And the reason they applied electrodes is, remember, I told you that early Earth is really stormy and really violent. Electrodes were the source of energy in the form of what they would have thought would have been lightning, while the only way to simulate lightning in their experiment was to use an electrode. Then they had a sea chamber, because remember, heterotroph hypothesis says that it's the primordial soup model, that this all occurred in water. So in that sea chamber, it provided some of the water vapor that was necessary. After allowing this simulation to run, they did end up discovering, after a period of time, amino acids at the bottom of the chamber. Now, amino acids are one of our biomolecules. It's one of our monomers. So they were able to produce a monomer in laboratory conditions that were pretty similar to that of early Earth. So that's one way you can support assumption number 1. Another way we can support the inorganic to organic is space. Now, this one we actually can see evidence of. The reason that they're willing, or scientists, biologists, are willing to say that there's potential that organic molecules came from space, in 1969 there was a meteorite that crashed to Earth, and we studied it. And in studying it, they found over 70 different amino acids. They found simple sugars on it. They found nitrogenous bases. So they found, your nitrogenous bases, adenine, thymine, cytosine, and guanine. They found all of those on this meteorite when it came to Earth. There's also been analysis of some of the gaseous remains of the tail of Halley's Comet. And in that tail material, they found formaldehyde. Formaldehyde. They found formaldehyde. And it's an important precursor to organic compounds. Formaldehyde would have aided in the formation of nucleotides if they hadn't been found elsewhere. So space here is actually legitimately a good potential hypothesis. The third possibility to support our first assumption is deep sea vents. Now, deep sea vents are exactly, what they sound like. They're hydrothermal vents on the ocean floor. We find them usually closest to volcanoes and shifting tectonic plates. Now, you should have just thought, well, shifting tectonic plates and volcanoes sounds a lot like what we've said about early Earth so far. And that's exactly why scientists find this a region worth studying, is that those places are very, very similar to what we think early Earth would have been like. They're very hot, and they're a source of methane and hydrogen sulfide. So they're hot and have some of those early atmospheric gases. And these bubble through that particular condition. Well, not very far away from those vents, the environment changes pretty drastically. And the temperature decreases a bit, and it becomes not quite as hostile. Near those deep sea vents, there's evidence, actually it's not even evidence, they found colonies of particular bacteria that we call archaebacteria. So you should be familiar with our archaebacteria from our classification chapter. Archaebacteria, remember we said archae or archaea means ancient, old. Archaebacteria live in these deep sea vents. They live here, they thrive here, and they're what one would call extremophiles. Now, it's kind of a funny name, but remember, the suffix phile, P-H-I-L-E, means to love. So an extremophile is a bacteria that loves extreme conditions. In other words, hot, reducing conditions, which would be exactly what you find at deep sea vents. Because there are living things there today, scientists theorize that there's possibility that that's where life could have begun. So that's where step one of our heterotroph hypothesis, there could very well be organic molecules that formed under these conditions. Now that we've supported the first assumption of the heterotroph hypothesis, we have to work on supporting the second assumption, how monomers could have, in fact, associated to make polymers. This support is actually a little bit simpler than the first assumption. This is only one hypothesis that we have that we think seems reasonable for how this could have happened. It's called the Cairns-Smith hypothesis. Again, we just name these things for the scientists who developed them. And the Cairns-Smith hypothesis says that there's a very special kind of clay. And this clay is created by weathering volcanic ash. I know, you're surprised, the word volcano came up again. So that weathering volcanic ash clusters into a clay. And that's what this top picture is intended on representing from your book. That clay clusters our monomers close together. And the closer chemicals are together, the higher the probability it is that they'll undergo a chemical reaction. In order to react with another molecule, you have to be close to it. The proximity has to be pretty small. So those, in this picture, you'll see that they're nucleotides that kind of cluster close to each other on this clay. The clay kind of serves as a spongy, porous surface that molecules can settle into. And once they've associated, or once they've settled in, they then react with each other. And when they react with each other, they go from being a monomer to a polymer. You'll notice here the polymer that we've made is a single-stranded nucleic acid. Most likely, according to scientists, this single-stranded nucleic acid would have been RNA. So it's essentially that clay acts as a substrate for the chemical reaction for the polymerization to occur. That polymerization, you guys are familiar with from the biochemistry chapter, as being the change from being a monomer into a polymer. So that's what allows all of those different bonds to form. Assumption number 3, I told you number 2 would be pretty quick. Support for assumption number 3, the polymers must self-replicate. We call this the RNA world hypothesis. Scientists in laboratory conditions have been able to create RNA that is able to catalyze its own replication. So in other words, they make a strand of RNA, and that RNA would have the ability to copy itself. That's the first time that scientists saw anything that would have been able to replicate, to reproduce. That catalytic RNA has a special name. We call it ribozymes. And it's just RNA that's able to catalyze its own replication. If this condition was met, if they were able to find a method that chemical evolution leads to a self-replicating molecule, then the conditions for creating life out of inorganic things would have been met. So that's the end of chemical evolution. That's what scientists believe would have been enough to give rise to very early life. So that's the end of chemical evolution for you.